Hello everyone, Namaste. Welcome back to my channel AcademicTuber. Today we are going to discuss last part from the unit Heredity, that is genetics. Before that, if you are new to this channel, please subscribe the channel, and for more updates hit the bell icon. If you want more videos related to this, please like and to share this video among your friends, and also don't forget to give your valuable response in comment section. Genetics. It is the branch of biological science which deals with the nature and behavior of genes. So we can also define genetics as the biological science of genes, heredity, and variation. The sub-branches of genetics are genetic engineering, medical genetics, etc. Gregor Johann Mendel was the first scientist to propose the law of genetics through various researches. Mendel was born on 22 July 1822 in Austria, and is also known as the father of genetics. Gregor Johann Mendel is known as the father of genetics because he introduced the concept of genes as the basic unit of heredity for the first time. He carried out many experiments on the pea plants grown in his garden to prove that hereditary characters transmit from parents to offspring. Heredity. It is the process by which living organisms transmit parental characteristics to their offspring. The another term for heredity is inheritance. We know that all the living beings can produce offspring like themselves. Due to this reason, they continue their generation by giving birth to their young ones. Offspring inherit the characters of the previous generation. Although the organisms may look the same, they have some qualities that differ from each other. Parental characteristics are transmitted to the offspring in both sexual and asexual reproduction. These traits are transmitted from one generation to another because of the genes present in the chromosome of the nucleus of the cell. Characteristic trait of pea plant selected by Mendel for his experiment. While doing experiment, Mendel considered 7 pair of contrasting characters in pea plants, which are: first one is height of plant, tall and dwarf; position of flower, axial and terminal; color of pod, green and yellow; shape of pod, inflated and constricted; shape of seed, round and wrinkled; color of flower, purple and white; color of seed, yellow and green. The characters denoted by capital alphabet are the dominant character, whereas the characters denoted by small alphabet are recessive characters. Why did Mendel selected a pea plant Pisum sativum for his experiment? Mendel chose or select pea plant for his experiment because of following reasons. First one, pea plants are bisexual and their flowers are closed, making them naturally self-pollinating plants. Next one, cross-pollination can be done if necessary. Their life cycle is short and offspring can be produced faster. They have many pair of contrasting characters. Many seeds can be produced at once, and due to this, many offspring can be produced. They are easy to cultivate. Because of these reasons, Mendel has selected pea plant for his experiment. Does Mendel law apply to human beings too? Is there any two difficulties Mendel would have faced if he had carried his experiment on human beings instead of pea plants? Obviously, yes. Mendel's law applies to human beings too. But while performing his experiment on human beings, many problems would arise if he had carried out his experiment on human beings instead of pea plant. He would have faced following difficulties. First one, in practical life, it would be difficult for him to cross between selected men and women for any two contrasting characters. They might not accept for crossing due to the social norms. Men and women don't have pure contrasting characters like that in pea plant, so he would have faced many problems to cross between any two contrasting characteristics. Mendel Experiment. Mendel studied 7 pairs of pure and hybrid traits found in pea plants separately and classified each offspring according to the character. He selected pure tall plant and pure dwarf plant and carried out pollination between them to study the heredity. Seeds obtained from that pollination were grown, which were called first filial generation. All the plants of first filial generation were tall. He performed experiments using the remaining pair of contrasting characters too. He found similar type of results. That is, only one character was expressed in each of the first filial generation when two pea plants each having a pair of contrasting characters were pollinated. The characters expressed in the first filial generation was called the dominant character, and the character which was hidden in the first filial generation was termed the recessive characters. Mendel carried out self-pollination between the hybrids produced in first filial generation. After self-pollination, the offspring obtained in the second filial generation were both tall and dwarf. Among them, 75% were tall and 25% were dwarf. Furthermore, while self-pollinating the pure tall pea plant of the second filial generation, all the offspring were pure tall pea plants. Self-pollination between the pure dwarf plants of the second generation produced only pure dwarf offspring. Similarly, self-pollination of hybrid offspring of the second filial generation yielded 75% tall and 25% dwarf pea plant, which is presented in the chart given below. Result of Mendel experiment. Tall plants were produced from pure tall plants. Tall and dwarf plants were produced in the ratio 3:1 from hybrid tall plants. Dwarf plants were produced from pure dwarf plants. So here are some of the important terms related to the genetics. First one is unit character. It is a definite external character of an individual of a crop. Each organism consists of its own type of character. Example, a rose plant has its own type of leaf, flower, stem, etc., which acts as unit character of it. Next one is allele. An allele is one form of gene. There may be two or more alleles of genes. In genetics, different alleles are denoted by letters. Example, capital T, capital T, small t, small t, etc. Next one is traits and character. Traits and character both are used to describe the quality of organism. Example, the blue color BB is a trait, but the eye color of an organism is a character. In this way, traits are conditions for a character. Next one is dominant traits. The traits which are prominent and appears in successive generation are called dominant traits. It is represented by the capital alphabet. Examples, in cross-pollination between tall pea plant and dwarf pea plant, tall is dominant trait because it appears in successive generation. Recessive traits. The traits which cannot express themselves due to the presence of dominant traits are called recessive traits. It is represented by the first letter of the word of dominant characters in small form. Example, in cross-pollination between tall pea plant and dwarf pea plant, dwarf is recessive character because it cannot express itself in successive generation. Next one is homozygous. When both alleles of a particular gene are identical, same, it is called homozygous. It is also called pure breeding. Example, capital T pure tall, small t pure drop. Next one is heterozygous. When all alleles of a gene are not identical, it is called heterozygous. It contains one dominant allele and other recessive allele. It is also called hybrid. For example, capital R, small r, hybrid red, capital T, small t, hybrid tall, etc. Next one is hybrid. Hybrid are the organisms produced after cross-fertilization between two genetically different organisms. The process by which hybrids are produced is called hybridization. Hybrids are of various types. Example, monohybrid, dihybrid, polyhybrid, etc. Next one is phenotype and genotype. The external appearance of an organism is called phenotype. It is expressed in words, example, tall, dwarf, etc. Whereas the genetic makeup of an organism is called genotype. It is expressed in letters, example, capital T for pure tall, capital T, small t for hybrid tall, small t for pure dwarf. Next one is F1 generation. The generation obtained by crossing two parental stocks is called F1 generation, and it is also called the first filial generation. Next one is F2 generation. The generation obtained by crossing two parental stocks of F1 generation is called F2 generation. Here in F2 generation, the offspring produced from the F1 generation are the parents. It is also called the second filial generation. Difference between monohybrid and dihybrid cross. Monohybrid cross involves only one pair of contrasting characteristics, whereas the dihybrid cross involves only two pair of contrasting characteristics. In monohybrid cross, as a result, offspring have phenotypic ratio of 3:1, whereas 3:1 in F2 generation, whereas in dihybrid cross, as a result, offspring having phenotypic ratio of 9:3:3:1 are produced in the F2 generation. Example of monohybrid cross is a cross between a tall pea plant and a dwarf pea plant. Here only one pair of contrasting character is involved, that is the tallness of a plant. Whereas example of dihybrid cross between a red flowered tall pea plant and white flowered dwarf pea plant. So here in example, it involves the two pair of contrasting character, that is color of flower and height. Difference between phenotype and genotype. Phenotype, it is the external appearance of organism, whereas genotype, it is the genetic makeup of organism. Phenotype, it is expressed in word, example, tall, dwarf, etc. Whereas genotype, it is expressed in letters, example, capital T for pure tall, small t for pure dwarf, etc. In phenotype, parental characteristics of organisms cannot be found, whereas in genotype, parental characteristics of organisms can be found. Next one is dominant trace and recessive trace. Dominant trace appears in successive generation, whereas recessive trace remain hidden in successive generation. Dominant trace suppresses the recessive characteristics, whereas recessive trace it is suppressed by the dominant characters. Mendel's law. There are three laws of Mendel's. First one is law of dominance, second one is law of segregation or purity of gametes, and third one is law of independent assortment. First one is law of dominance, which is also known as the first law of Mendel. It states that when a cross is made between a pair of pure contrasting characters, only one of them is able to express itself in F1 generation, while other remains hidden. According to Mendel's law of dominance, when a cross is made between two pure individuals having a pair of contrasting characters, only one character is expressed externally in F1 generation, which is called the dominant character. Hence, the character or the trace which is expressed externally in F1 generation is called dominant character, and the character which remains hidden in F2 generation is called recessive character. When a cross is made between a pure tall and pure dwarf pea plant in their F1 generation, all the pea plants are seen hybrid tall. Here tallness appears by suppressing the dwarfness. In this cross, tallness is the dominant characteristic as it appears in F1 generation, whereas dwarfness it is recessive characteristic as it remains hidden in F1 generation. Here we can see in diagram as well. All the offspring they contain both the characters of tallness and dwarfness, but dwarfness it is not able to show or express itself in successive generation, whereas the tallness, which is the dominant character, uh, express itself in F1 generation. Although there are two characteristics, that is tallness and dwarfness, only tallness is seen because tall is the dominant characteristics. Next one is the law of segregation, which is also known as the law of purity of gametes. It says that the hybrid obtained in F1 generation contain two contrasting characters which remain together for a long time but not mix with each other and separate or segregate at the time of gamete formation. Thus, any gamete is not impure. Although two different characters coexist in the hybrid of the first generation, they remain pure without losing their originality. In the formation of gametes in the hybrid during meiosis cell division, the genes of the pure or hybrid allele in the mother cell are separated and only pure characters enter each gamete. It means gametes formed are pure. That's why this law is known as the purity of gametes. In F1 generation, all pea plants becomes hybrid tall with dominant capital T and recessive small t characteristics. Both characteristics remain together for a long time without mixing and finally separate into two different pure gametes, either tall or dwarf. These hybrids after interbreeding produce three different types of offspring, that is pure tall, which is denoted by capital T, hybrid tall, which is denoted by capital T small t, and pure dwarf, which is denoted by small t in the ratio of 1:2:1 in the F2 generation. It is possible due to segregation of gametes. Monohybrid cross in guinea pig. When a pure black guinea pig is crossed with a pure white guinea pig in F1 generation, all the guinea pigs are found black, as black is the dominant character. When the guinea pig of F1 generation are crossed together in F2 generation, 75% guinea pig are found black and 25% are found white. In this case, phenotypic ratio of black and white was 3:1 and genotypic ratio was 1:2:1. Genetic technology. It is the process of modifying genes by understanding genetic expression, taking advantage of natural genetic variation and transferring genes to new organisms. Currently, the world has advanced a lot in the field of technology, and genetic technology is one of them. This technology helps to develop new qualities by making various changes in DNA easily and quickly. In this technology, modification in the genetic material or gene is carried out. Nowadays, scientists have been able to find out what genes are, their function, and how they can be altered by adding, deleting, and substituting DNA. Genes have coded instructions that are used to synthesize protein and to transmit the hereditary characteristics. Among the various technologies, genetic engineering is the process that alters the DNA structure of an organism using laboratory-based techniques. It is also called genetic modification. In this process, a new trait is developed by changing a single nitrogen base pair, deleting or adding a gene in the DNA. Gene variants created through genetic engineering can be passed from one generation to next. Identifying genes and their function is an important application of genetic technology. Genetic technology has an important place in biotechnology and molecular biology. It has made it possible to change and regulate the plant characteristics by using DNA sequencing information in its structure, function, process, etc. These modern DNA technology with high potential and need are the important ways to guarantee the sustainable development of agriculture. Nowadays, DNA technology is mostly used in the field of forensic science. This technology is used to investigate various criminal cases called the DNA case. There are three main categories of concern regarding the application of genetic technology. First one is cloning, the creation of either embryo or babies that are genetically identical to their living or dead parents. Next one is trait selection, selection of sperms, egg, or embryos that possesses genes which are associated with certain traits. And third one is genetic modification, changes and manipulation of genes in living cells. Role of DNA tests in different researches. First one is forensic. Forensic team often use DNA to determine if a person is a suspect or if they should be eliminated as a suspect. It proves a person's innocence or guilt, or at least it can prove whether a person was in the vicinity of the crime scene. Next one is paternity test. DNA extraction is also helpful for determining the parents of the child. Third one is ancestry tracking. DNA extraction can also help a person understand the places their ancestors further back came from. Next one is medical test. DNA extraction is helpful in identifying if a person is a carrier of any of the diseases. Next one is genetic engineering. DNA extraction can be helpful for genetic engineering of both plants and animals. In plants, it is helpful in identifying, isolating, and extracting the wanted genes to replicate in successive generations of plants, whereas in animals, cloning animals to transferring one animal's DNA to another. Next one is vaccines. DNA vaccines are often used in various animal vaccines and general development of some human vaccines. Next one is hormones. DNA extraction helps in the production of human growth hormones and insulin through recombinant DNA technology. So here is one question. What should be done to make DNA testing reliable, fair and effective? To make the DNA testing reliable, fair and effective, special attention is required during the collection and transportation of samples. Samples, it may be hair, skin or blood. Samples should also be protected from contamination. Next one is selective breeding. Selective breeding, it is the process of selecting animals and plants with the best qualities and interbreeding them to produce offspring with desired characters. Since ancient times, peoples have been selecting and breeding plants and animals with good qualities for agricultural products they want. In this way, the main purpose of selective breeding is to introduce the desired traits in an organism and establish those traits in the future offspring. It involves selecting and breeding the mother and father or both to produce desired plants and animals. It emphasizes natural reproduction using the gene variation occurring naturally in organisms. For example, if we reject dwarf animals and breed only tall animals, the offspring inherits the gene for tall. If this process is repeated for many generations, the tallness traits will be established in future generations while the quality of being dwarf or short will disappear. So here is one question. Why do some people criticize selective breeding? Some people criticize selective breeding because during selective breeding, some natural qualities may disappear or can be muted and undesirable qualities may appear in animals. Disadvantage of selective breeding. It increases the population of plants and animals having similar genetic traits. There is a chance of spreading infectious diseases genetically. It increases the possibility of suffering from hereditary diseases as genetically close organisms are used in the reproduction. It is also called artificial selection because it involves human interference and it inhibits some naturally occurring genetic traits and can affect biodiversity, making it possible for species to become extinct in the future due to some bad traits. Method of selective breeding. First one is inbreeding. It is a method in which closely related animals are allowed to interbreed. Organisms produced in this way are described as pure breed or inbreed. Next one is line breeding. Line breeding, it is a type of inbreeding in which breeding is done between more distant relatives to get animals with desired characters. It also reduces the risk of ill health that can sometimes be seen on pure breed animals. Next one is crossbreeding. It is a type of breeding which is done between two different species of same genus. This method is suitable to produce offspring which display the characteristics of interest by crossing two pure breed organisms. Offspring produced by crossbreeding are called hybrids. The main purpose of crossbreeding are to enhance the quality of hybrid. Next one, to take the advantage of heterosis, hybrid vigor, to use average breed effect, to design a cow herd, to target markets, to create breeding plans for a herd, etc. Some organisms produced from crossbreeding. First one is liger. It is a hybrid animal obtained by crossing the male lion and female tiger. It is larger than its parents and behave like lion. Next one is tigon. It is a hybrid animal obtained by crossing the male tiger and female lion. It is smaller than liger and either of its parents. It resembles a tiger, but most qualities such as roaring and socialization are like that of a lion. Next one is buffalo. It is a hybrid produced by the cross between a buffalo, American bison, and a bull. They are different from other hybrids because they can reproduce. Next one is zebroid. It is a hybrid animal obtained by crossing zebra and horse, and they cannot reproduce. Mule, it is a hybrid animal produced by crossbreeding of donkey and a horse, and it is sterile, that is infertile. Next one is pomato. It is a plant produced by crossing potato and tomato, and in this plant, tomato is produced in the stem above the soil and potato is produced underground. Advantage of crossbreeding. This method combines the desired qualities of two organisms from different breeds, varieties, or species. Human beings can produce organisms with desired characters. Crossbreeding helps the opportunity to make full use of a wide range of genetic material. Animals that exhibit better quality than the parent animals can be developed. Immunity, strength, age, vigor, etc. of an organism can be improved. Crop production can be increased from plants produced by this method. Next one is disadvantage of crossbreeding. There may be a policy problem in the future in the lack of right understanding and management. The price of the product obtained from crossbreeding is lower compared to that of the products obtained from pure breed, so farmers can't earn as much as expected. There is a limitation in the sale of animals produced by crossbreeding in the export market. In many cases, it becomes a cause of extinction in original animals. So, differences between selective breeding and crossbreeding. Selective breeding, breeding animals or plants with specific traits to amplify those traits over generation, whereas crossbreeding, it is the breeding of two different breeds or species to combine desirable traits. The main goal of selective breeding is to enhance specific traits or characters, whereas the main goal of crossbreeding is to combine desirable traits from parent breeds. Parent selection. Selective breeding based on the desired traits, whereas in crossbreeding based on the compatibility and complementarity. Genetic diversity may lead to reduced genetic diversity over time in selective breeding, whereas in crossbreeding it introduces genetic diversity by mixing traits. Complexity in a case of selective breeding is less complex, whereas in crossbreeding it is more complex. Selective breeding can be a relatively long-term process, whereas crossbreeding it is we can obtain the result very quickly. Next one is artificial insemination. Artificial insemination. Artificial insemination it is a technique of collecting semen from advanced breeds of a male and allowing them to enter the female reproductive tract at the right time through the use of equipment. Nowadays, because of development of various technologies, fertilization is possible without mating between male and female organisms. The practice of producing offspring of advanced variety has greatly increased nowadays, which is by collecting semen from the male located at far distance and inseminating into the body of the female organism. The first scientific research on artificial insemination of domestic animals was carried out in dog in 1784 by the Italian scientist Lazzaro Spallanzani. The main objective of artificial insemination is to produce a large number of advanced offspring by transmitting the sperm of an advanced breed of male to a female ready for conception. Currently, artificial insemination is in practice instead of natural mating in many animals like cows, buffaloes, goats, sheep, etc. to improve genetic quality in animal husbandry. So here is a diagram showing the artificial insemination in the cow. Artificial insemination helps to strengthen farmers' economic status. With this technique, the farmers in animal husbandry can avoid the trouble of rearing expensive males with special qualities. Advantage of artificial insemination. There is no need of rearing male for breeding, which saves expenses of rearing. It helps to control the infection and spread of diseases during mating. After collecting semen from the male, it is tested and fertility is checked to ensure the fertility of male. Collected special semen can be used even after the death of male. Collected semen can be easily transported over long distance for fertilization. This method helps to prevent injury in the male or female at the time of fertilization. This method increases the rate of fertilization as well, and it helps to keep a good record of reproduction. Although it has lots of advantages, it also has some disadvantages too. It needs a well-trained manpower and special equipment. It requires more time than natural breeding. Reproduction may not take place, or there might be a chance of infection if the equipment is not properly sanitized during artificial insemination. Next one is in vitro fertilization, IVF. It is the process of fertilization where an egg is combined with the sperm in vitro in glass. It is a type of assisted reproductive technology used for infertility treatment and gestational surrogacy. This method is a complex series of procedures to help couples with reduced fertility and genetic problems. The process involves monitoring and stimulating a woman's ovulatory process, removing an ovum or egg from her ovaries and letting sperm fertilize them in a culture medium in a laboratory. In IVF, a mature ovum from the ovary of the female is taken and stored in a petri dish and fused with the sperm of the male in a sophisticated laboratory, and the fertilized ovum is then transferred to the female's womb uterus after few days. IVF can be summed up in the several steps. First one is ovarian stimulation, after that egg retrieval, after that sperm retrieval, after that fertilization and embryo transfer. It takes about 3 weeks for a complete cycle. The embryo grows in the woman's uterus as in normal pregnancy. So here is a diagram with a series of steps involved in the in vitro fertilization. First one ovarian stimulation, hormone therapy. Second one egg pick up. Third one is sperm preparation. Fourth one egg fertilization. In fifth there is embryo development, and finally embryo transfer. Advantage of IVF. IVF makes it possible for the couples who are unable to bear a baby to have a baby. It allows conception by using the couple's own sperm and ovum or by the use of donor sperm and ovum. It is more successful than other assisted reproductive techniques. It is helpful to solve the problems related to various chromosomal disorders in the child. Infertility and genetic problems can be solved by this technique. It increases fertility and reduces the risk of miscarriage as well. It increases the chance of having a healthy child. Disadvantage of IVF. There is no guarantee of a successful IVF cycle. It may not be successful and may take more than one cycle as well. Sometimes it shows the problem of multi-fertilization. It is an expensive method. Adopting this technique may cause emotional stress in the couple. There is also a chance of premature birth of a baby with low weight. So difference between artificial insemination and in vitro fertilization. Artificial insemination is the introduction of sperm into the reproductive tract through methods like the catheter or syringe, whereas in vitro fertilization it is the fertilization of eggs and sperm outside the body followed by transfer of embryo into the uterus. Artificial insemination location it is inside the female reproductive tract, whereas in vitro fertilization where the fertilization it is outside the body in the laboratory. Fertilization site. Artificial insemination fertilization site it is in the fallopian tube or uterus depending on the method used, whereas in vitro fertilization site it is in a controlled laboratory. Artificial insemination sperm donor may be used, whereas in vitro fertilization both sperm and egg donors can be used. In artificial insemination, egg retrieval it is not involved, whereas in vitro fertilization, egg retrieval egg are retrieved directly from the ovaries. Embryo development occurs in the female's body in artificial insemination, whereas development occurs in the lab in the in vitro fertilization. Embryo selection in artificial insemination is limited control over control, whereas in vitro fertilization embryo selection there is better control over selecting the viable embryos. So here are a few reasoning questions from this unit. DNA testing is a reliable technique for criminal investigation. DNA testing is a reliable technique for criminal investigation because each individual's DNA is unique, making it highly accurate for identifying suspects and linking them to the crime scenes. Next one, genetic engineering involves the detailed study of DNA. Why? Because DNA contains the genetic information that determines an organism's traits and characteristics, and modifying this information enables the manipulation of those traits for a specific purpose. Next one, crossbreeding also result in sterile offspring. Why? Crossbreeding can result in sterile offspring due to the phenomenon called hybrid sterility, where genetic differences between the pure parent species lead to the reproductive incompatibility in the hybrid of the offspring. Next one, special attention should be given while collecting the samples of DNA testing. Why? to ensure accurate and reliable results. Contamination, mishandling, or improper selection can compromise the integrity of the DNA and lead to incorrect conclusions. That's why special attention should be given while collecting the samples for DNA testing. So here is definition of DNA testing. DNA testing, it is a scientific method that involves analyzing an individual's DNA, the genetic material that carries unique information about their ancestry, traits, and potential health conditions. And lastly, here is the difference between inbreeding and outbreeding. Inbreeding, it is breeding between closely related individuals within the same breed or lines, whereas outbreeding, it is the breeding between unrelated individuals from different breeds or population. Inbreeding reduces genetic diversity by increasing the likelihood of inheriting identical alleles, whereas outbreeding increases genetic diversity by introducing new alleles from different sources. Inbreeding involves mating between close relatives like siblings or cousins, whereas outbreeding involves mating between unrelated individuals. Inbreeding can amplify both desirable and undesirable traits. Outbreeding, it can result in masking of undesirable traits. Inbreeding generally though as does not benefit from hybrid vigor, whereas the outbreeding can benefit from a hybrid vigor, leading to healthier and more vigorous offspring with high immunity, less chance to be caught by any sort of diseases, etc. By this, we have completed the note of this unit. I hope this video was useful to you. If you like this video, please share with your friends and also don't forget to subscribe. If you have any queries, drop the comment in the comment section. See you on the next video. Thank you.